Alright, so here we are. I don't know what I was talking about before in Lecture 19, but this is the real Lecture 20, and we will wrap up photosynthesis as well as wrap up this course in undergraduate biochemistry. Can't believe we're already here. This is the second half of Chapter 22. This is going to be a two-chunk lecture, so we'll only be dealing with Lecture 20A and Lecture 20B. In this first chunk, we're going to double back and briefly review the light reactions so that we start from the same page, make sure that we understand what we're going to be feeding into the dark reactions today, and we'll set the stage for the dark reactions and talk in general about how we fix carbon dioxide to make sugars. The details of the dark reaction, which essentially boil down to a process called the Calvin cycle, our last cycle of the course, will be covered in the second chunk, as will a little bit of material about how tropical plants concentrate carbon dioxide. Carbon dioxide is easily lost in very hot weather, and so we'll talk briefly about how tropical plants concentrate carbon dioxide for their own photosynthetic purposes. But to move on and kind of, again, double back and briefly review what we've already talked about, we know that photosynthesis in general is occurring in the organelle of plant cells, which we refer to as the chloroplast. And what happens in the light reactions is that water is absorbed by the plants, and energy from sunlight directly is used to oxidize that water, strip off the protons, the electrons, and release molecular oxygen as a byproduct. And that's really what Lecture 19 was all about, talking about how light energy is absorbed by antenna chlorophyll molecules, how oxygen is released at reaction centers. Remember, we did absorb sunlight solely to oxidize water, and that oxidation released electrons, protons, and oxygen. But the plants were not interested in the oxygen at all. The plants wanted the electrons to load onto NADP to serve as those high energy, high impactful reducers. And the plants wanted the protons as well to create a proton gradient that could then be used to power an ATP synthase-like enzyme and produce ATP, all, again, from the energy derived from sunlight. We want the oxygen. It's us who benefit from the side effect of the light reactions. The oxygen is waste for the plants, but the oxygen, of course, is our ultimate electron acceptor. We could not do aerobic respiration without that oxygen released by green plants, and so very much the plant's waste is our benefit. That means that the plant's trash is our treasure, of course. We could not survive on this planet without this action of green plants. So in general, plants use the electrons that they harvested from water to pass what is really stored light energy. When we charge up those electrons in photosystem 2 and photosystem 1, we are capturing within, or as part of those electrons, directly energy from the sun, sunlight energy. And so by passing those electrons around in the chloroplast, cells are quite literally passing around sunlight energy. And we talked quite a bit about this diagram in general, the energetics of the light reactions. We have water arrive at the chloroplast. It is instantly and immediately oxidized by sunlight energy by this reaction center, photosystem 2. And when those electrons are harvested from that oxidation reaction, they are charged by sunlight energy, photosystem 2 responsible for that charging. Unfortunately for the plant and for photosystem 2 is unable to charge those electrons to their full capacity, and so those electrons must be passed on to photosystem 1. But whenever you pass electrons, you're going to lose some energy along the way. And so those electrons are transferred by an electron transport chain to photosystem 1, but they do kind of lose some energy as delta G's along the way. Negative delta G's. But that's okay, because photosystem 1 can make up the difference and charge those electrons some more and get them to their full charged capacity, get them to where we fully need them to be, so that they are then ready to reduce NADP plus and make NADPH. All of the protons that were stolen from water along the way are also used to create a proton gradient. That's what the plant does once it has achieved as many high-power electrons as it needs. And so those protons create a proton gradient, and they flow through an ATP synthase-like enzyme, making ATP. And they do that very much as we described for oxidative phosphorylation in our mitochondria. In fact, the only thing that differs between oxidative phosphorylation and the process in plants is where the initial energy comes from. For us, it comes from redox reactions, that's why it's oxidative phosphorylation, and from plants it comes from sunlight, and so we call it photophosphorylation. Plants are also making this NADPH super reducer along the way as well. The ATP production is secondary. It's the NADPH that plants want the most. It's the super reducer they need. So then the question becomes, well, why do plants need a super reducer? Seems like a whole lot of work to make NADPH. What's the whole reason for that? And you may remember this from the last lecture. I know there was quite a bit going on in the light reactions, and I fully appreciate that, but perhaps you recall that we said carbon dioxide is fully oxidized carbon. It has been milked of all its high-power electrons and then released by animals like us back into the environment as waste. So the carbons in carbon dioxide are fully energy depleted and they are fully oxidized. If we're going to take that carbon from carbon dioxide and use it to make sugars, we need to reduce that carbon. If it's fully oxidized, we need to revert it to a reduced state. And in order to reduce fully and very oxidized carbon dioxide, we need a super reducer. So the extensive reducing power of NADPH is what we rely on to reduce the carbon of carbon dioxide and get it to take on protons and electrons. To hydrate those carbons into carbohydrates, we need a super reducer. So that's where we're going in all of this. Let's fix some carbon from carbon dioxide into glucose sugar. Let's do these dark reactions and make sugar in a plant cell using nothing more than carbon dioxide, ATP, and NADPH. That's what the dark reactions allow. Additionally, we should keep in mind that it's not as though the plants need this glucose they're about to make in the dark reactions right now. Photophosphorylation has created tons of ATP for the cells to live off of. So everything that the cells do in the dark reactions is used for energy storage. This is glucose that the cells are going to make with surplus light energy that they can store as starch and use for a cloudy day, if they happen to fall into the shade. So plants that can't do the dark reactions are still around, or at least as algae and lichens are still around on our planet, but they can only make energy in the here and now. They can't survive long periods without direct sunlight. More modern green plants can, because they make this energy, they make this stored glucose energy in starch. So keep in mind that the plants are not hurting for energy. If the sun was shining, they made plenty of ATP through photophosphorylation. It's the long-term energy that they are now investing in. The dark reactions make glucose that can be stored as starch so that energy can be produced later if necessary. Incidentally, plants also need a lot of glucose in order to build all their cell walls and make all that cellulose. And glucose made by the dark reactions is also used for cell wall production in green plants. Everything we talked about in the last lecture was occurring either within those thylakoid membranes of the thylakoid discs, or in the lumen of the thylakoids, within that thylakoid space. Here, the dark reactions actually occur in the stroma, the open space that lies outside of the grana, but still enclosed by the inner mitochondrial membrane. So we're kind of switching our location in the chloroplast as well. And finally, what we are about to do is essentially take 6 carbon dioxides, because there are 6 carbons in glucose, take 6 carbon dioxides and hydrate those carbons as a chain to make one molecule of hydrated carbon glucose. So take 6 carbon dioxides and make one glucose molecule from it. The only problem is that's going to take a whole lot of high-power electrons, so we're going to burn through 12 NADPHs, creating 12 NADPs, oxidized. And we're also going to use 18 molecules of ATP and burn those to 18 ADPs and their inorganic phosphate. Now that's okay, because we made plenty of ATP, and NADPH through the light reactions. So the cells have these things to spare. We're investing in the future. We're taking an energetic hit and burning through a lot of NADPH and ATP in order to make glucose for a rainy day, literally for a rainy day. So that's where we're heading in the next chunk. In the next chunk, we'll tell the full story of the dark reactions, biochemically, of course. But here, all we were really interested in knowing was that we are going to make glucose. We're going to make reduced carbon glucose using the dark reactions. We're going to use ATP and NADPH that we harvested from the light reactions, and we are not doing it for immediate energy right now. We're doing it to make glucose that can be used either for energy storage in the form of starch or to make cellulose for those plant cell walls. So moving on to chunk B, we'll see exactly how these dark reactions work.